Hey everyone, welcome to Professor Long's Lectures in Microbiology. I'm Professor Bob Long. These videos are intended for use by students who are enrolled in my courses. If anyone else out there in YouTube land finds them helpful, by all means, please hit like and subscribe or let me know. If you don't like them, let me know. I will tell you that these videos are done very crudely with a cell phone, me, and a board. They're done in a very short period of time in order to meet deadlines under this coronavirus situation that we're in, having transitioned from face-to-face to online courses. Now, I do hope to reshoot them in the future and make them much cleaner and neater and more concise. So forgive me if I ramble or if I stumble over my words a little bit, because I wasn't intending to do this way. Nonetheless, also they're only to provide a fundamental and foundational understanding of some of the basic principles in microbiology. So the details we can add over time. So if your instructors in other courses go into more detail, then you need to learn what they want you to learn. These are intended for my students. Now, we've been talking about, we've begun, in, at least in our textbook, I believe it's chapter 14, where we're talking about diseases and epidemiology. In the last video, we did a whole bunch of definitions, and we talked about the normal or resident flora. And we talked a little bit about contamination and disease and infection. Now we're going to go a little bit more into diseases, how we detect disease, how we describe diseases, some things that can affect the occurrence of disease within a population. So one of the first things we want to talk about is how do we sort of find diseases or discuss diseases, or finding the cause of a disease. And really one of the first guys was the scientist Koch, K-O-C-H, and they are called Koch's postulates. Okay, oops. So to postulate means to hypothesize, and Koch's postulates basically say this: one, the same microbe must be present in all cases of the disease. Let me rewrite this. My penmanship, I need to work on it a little bit on these marker boards. I can write much neater, but I'm usually trying to hurry to get a lot of information in a short period of time. But whenever you have any case of a disease, if you have 20 cases of the disease, but in 3 of them, the same microbe is not present, it's hard to say that this microbe causes that disease if it's not always there. So every time you have a case of the disease or an instance of the disease, the same microbe must always be present, okay? Now, the pathogen must be isolated from the diseased host. Okay? You actually have to be able to take that microbe out and isolate it, okay? And in this situation, we have to be, also must be grown in pure culture. So we have to know that in every instance, in every case of this disease, this microbe is present. We can isolate the pathogen and grow it in pure culture, where it's the only organism growing, and therefore we can say, well, we know that this organism must have come from that diseased host. A new host must develop the disease when inoculated. Or you should be able to inoculate the pathogen into a new host and still get the same disease. So any new host must develop the same disease when inoculated. And I put developed. Okay? So I know this organism is present in every case. I can isolate it from a diseased host and grow it in pure culture. I can take the pure culture, inoculate it into a new host, an organism that did not have the disease before, the same organism possibly, not the same one, but the same type, and I should be able to develop the disease. Finally, the pathogen must be isolated from this new diseased host. Okay? So, I know all these organisms have the same disease. These hosts all have the same disease. I should be able to isolate, or I must know that this microbe is present in everyone that has the same disease. That microbe should be able to be isolated and grown in pure culture. I can take the cultured microorganism, introduce it into a new healthy animal, and get that disease again, and then isolate that disease from that animal by saying, and that tells me I've taken it from something that was diseased, I've purified it and made sure nothing else was present. I put it in a new healthy animal and got the same disease, and I was able to isolate that organism from that diseased animal. And therefore I can say that this particular organism is the causative agent of a specific disease. This pathogen causes that disease based on Koch's postulates, okay? So you should know Koch's postulates. You should know them in order. Some people break them out in 5 steps or 4 steps, but nonetheless, it basically breaks down to this. Same pathogen is present. The pathogen must be isolated and cultured. The pathogen must be inoculated and cause disease, and it should be isolated again from the new diseased host, okay? So now there are a couple of limitations to Koch's postulates. For example, there are some diseases that only occur in humans. Well, if I was going to try to run Koch's postulates on humans, this would be considered medically and morally, and it would be unethical for you to give a disease to another human being that didn't have it, especially if that disease is very harmful. So if a disease is unique to humans, then we can't really run Koch's postulates. This works great for like smallpox or cowpox, but it doesn't work for certain diseases. Some pathogens, by the way, also can cause several different diseases. That makes Koch's postulates a little bit difficult to execute as well. And there are a couple of microbes in the world that we have not been able to isolate in pure culture. One of them is syphilis. We simply can't grow it in pure culture, which leads some to question it, but we know that syphilis is present in every case of the disease, and it will cause the disease if introduced into a new host. Syphilis being sometimes a sexually transmitted disease. There are certain viruses that cannot be grown in pure culture. So we can't really, study those viruses in a great deal. So that's Koch's postulates, and those are the limitations of Koch's postulates. Now, that was sort of at the beginning of microbiology and the beginning of the understanding of microbes and pathogenicity of microbes. A few other things that we need to talk about. When we talk about disease, obviously we're talking about the definition we used before, which is a disturbance in homeostasis or a disruption to the homeostatic condition of an organism, or the disruption of health. Okay, so a disease is nothing more than a disruption in the health of an organism. Now, when we talk about diseases, we need to talk about how do we know one is present. One of the things that we can talk about is signs and symptoms. And sometimes you'll see this on some reports. What are the signs and symptoms of disease? Well, what do people mean by that term? Well, when we talk about a symptom, we talk about a disturbance in a healthy condition as described by what we would say is the patient or the person experiencing the illness. And these are, what's a real big key to these are subjective. Now when we talk about subjective means it's sort of an opinion, or it's your experience or your sense of something. Objective means it is or it is not. For example, when I talk about subjective, pain might be a symptom of some kind of illness, like a headache. But I can't measure your headache. What I can do is I can ask you to describe your headache on a scale of, say, from 0 to 10, or 1 to 10. 1 meaning no pain, 10 meaning you need something now. And so doctors often, and medical care professionals often, ask someone to describe their symptom based on a scale of 0 to 10. And that would be a symptom. So it's some kind of disturbance in the health condition, but it's described by the patient, and it is subjective. It's not really measurable, okay? A sign, oops, I wrote seen. When we're talking about a sign, we're talking about a measurable objective disturbance in health. Or in the healthy condition. Okay. So, symptoms are some kind of change in body function that we describe sort of subjectively, but the key to this one is that these are objective. They can be measured. For example, your fever, your body temperature. I can tell you if your fever is low grade, if it's like 99.5 or 101.1, or a high grade fever, like 104.5 or something. We can measure that. So we can measure certain signs. We cannot measure symptoms. I can only ask the patient to describe it, and those descriptions are subjective, okay? If I were to try to give you a couple of examples of some symptoms, I'll come back to signs in just a moment. Now, for example, one symptom might be something that we call paresthesia. When we talk about sthesia, we're talking about how we feel, how we determine something. So paresthesia might be described as like a tickling sensation, prickling sensation, a numbness, a lack of feeling, okay? So when we're talking about paresthesia, this would be some kind of, would be a symptom. I can't measure, or we might say it's also like numbness or a burning sensation, okay? So you might say, man, it really hurts, like my skin feels like it's on fire right here, or I feel like there's a prickling pain or a tickling pain. Like prickling pain would be like if you hit your funny bone and you feel all the little picas hitting you, okay? Well, I can't measure that, but you can describe that. And those are called paresthesias, tickling and prickling and numbness or burning sensations to a part of the body. Another example of a symptom might be something like nausea. I can't measure a patient's nausea, but I can ask you to describe it. Do you feel a little nauseous? Do you feel very nauseous? Do you feel like you're going to throw up? And nausea is just nausea, like the desire or the need to vomit. And we can talk about what is called malaise or general malaise. Well, if you speak any Spanish, malo means bad, so malaise means a bad feeling, not feeling well. A tiredness, weakness, lack of energy. So when a patient comes in, like when you have the flu, you just don't feel well. You just know I don't feel well. You may not be able to describe it. So in medical terms, we might say that someone has malaise or general malaise. You might describe someone's nausea, or do they have some kind of paresthesia? Those would be symptoms. So those are disturbances in the healthy condition, but there are subjective observations. They are not measurable. When we talk about sign, again, signs are objective and they are measurable observations in a disturbance in health. Okay? When we talk about a sign, and let me just erase this so I have the word sign already written here. I can talk about things like blood pressure. I can talk about temperature. Okay? I can measure your blood pressure if it's gone up or down. I can measure your temperature. I can do things like, I can measure pyrexia, which is your fever, or your temperature. Pyro, meaning like fire, so pyrexia is a temperature or your fever. I can measure things like edema, how much swelling there is. We can observe it, we can see it, we can actually measure it. I can measure a spot. I can, erythema, okay? We're talking about redness to a tissue, so I can see how red a tissue is. Is it just a little red? Is it very red? I can measure the area of redness. Sometimes if someone has like a small infection that's developing around a wound, you will literally take a Sharpie and draw a circle around the area where the redness is. And you observe if the redness starts to creep outside that circle, then you know the infection is spreading and you might need to start prescribing antibiotics. So we can measure erythema, we can measure edema, we can measure temperature or pyrexia, we can measure blood pressure, okay? We can also measure changes in blood count. And when we talk about this, there's a number of terms that we can talk about. When we talk about cytosis, we're talking about an increase in the number of cells. So if I were to talk about erythro, and I throw this term out there, I put these two words together. Erythrocytosis means an increase in the number of red blood cells. If I were to talk about a leukocytosis, leuko referring to white blood cells, then I would have an increased white blood cell count. That could be a sign of an infection, or at least a sign of some kind of allergic response, some kind of immune response. If I talk about the term penia, that means a decrease in cell numbers, or a decrease in something. So if I talk about erythropenia, that means a decreased red blood cell count. Since red blood cells carry oxygen, you might feel lethargic. Leukopenia would be a decreased white blood cell count. Why would your white blood cell count go down? That would make you susceptible to other infections because your white blood cells are your immune system. So these are all signs. These are all measurable, and we can talk about them. All right, we can make objective observations of them. Symptoms are not objective, they are subjective, and they're described by the patient. Signs are measured by someone other than the patient. Now, one last term I want to talk about when I talk about this is I want to talk about syndrome. What is a syndrome? When we talk about syndromes, there are a number of diseases that have syndrome in the name. AIDS is acquired immunodeficiency syndrome. SARS is sudden acute respiratory syndrome. And when I'm talking about a syndrome, we're talking about a group of signs and or symptoms that are common to a specific disease or illness. So there are a number of signs and symptoms that always occur with this particular disease, and so we can say that that is a syndrome. For example, in AIDS, acquired immunodeficiency syndrome, one of the particular signs that we see is a decreased white blood cell count, particularly what are called T cells. People can also start to experience things like fevers, swelling of the tonsils or lymph nodes. You can start to experience things like rashes and a host of other flu-like syndromes and pneumonias. They always seem to occur with this particular illness, and so we would call that a syndrome. So sudden acute respiratory syndrome always has things like fever, shortness of breath, difficulty breathing, fluid in the lungs, and pneumonia developing. SARS, sudden acute respiratory syndrome, belongs to or is part of the same family as the COVID viruses. There's SARS-CoV-2, there's COVID-19, and they all result in a sudden acute respiratory, or they could result in a sudden acute respiratory syndrome. We have MERS, which is Middle Eastern Respiratory Syndrome, and a few others. Okay, so anytime you hear syndrome, it just means that there's a whole group of signs or symptoms that are common to that specific disease. Okay, now, excuse me, my eye is itching and I need a drink of water. Now, when we go to describe diseases, there's a number of ways that we can describe diseases, and they are described based on a number of factors, okay? We said before that epidemiology is the study of the occurrence of a disease and its impact on a population. So epidemiology really is the study of the causative agents, the etiology, the pathology, how the illness is created by a particular disease or pathogen within a population, and the impact on the population. How does it spread within the population? What is the mode of spreading? Is it latent or whatnot? So when we're talking about the epidemiology, we're talking about the study of all the parts of a disease, how it is acquired, how it is spread, and what impact it has on a population. And again, we said etiology or etiology is the study of what caused the disease. What is the causative agent? Now, when we talk about diseases, one of the things that we can describe them as is on how they are spread. So when we talk about a disease as being communicable, that's like communicating, interacting with others. A communicable disease is a disease that is spread to humans. Okay, it can be communicated, and when we're talking about, when we're talking about in the health sciences, this is particularly what we're discussing, is a disease that can be spread or carried to a human to a human being. Now, when we talk about a communicable disease, we can say whether that communicable disease is contagious. If it is contagious, that means it can spread from human to human. Okay? A communicable disease is any disease that is spread to humans, and when we say that it's contagious, that means anyone who's infected could spread it to a non-infected person from human to human. And examples include things like the flu, or we could talk about COVID-19, we could talk about sexually transmitted diseases. Those are contagious and communicable. Pink eye, conjunctivitis, is a communicable disease if it's caused by a particular bacterium. Now, this is usually done through direct contact with people, by exchanging body fluids, kissing, sexually transmitted diseases, intravenous drug use and sharing a needle with people, or someone not using a sterile needle in a hospital. That's why you throw everything away, you can't reuse things. That's to stop the spread of communicable diseases. Now, I'll stop there. So when we talk about non-contagious diseases, these are diseases that are not spread from human to human, okay? But we can also talk about a non-communicable disease. Okay? So when we say that a disease is non-communicable, that means it cannot be transmitted. From a diseased person. Okay? So, for example, you might get food poisoning. And if you get food poisoning from botulism or something and you're puking your guts up, someone else who comes around you is not normally going to get food poisoning from you. You could sleep right next to them. You can sleep with your child that's gotten food poisoning and not get sick because it's not a communicable disease. It's not going to spread. It's not contagious, and it's not even a disease that's usually, well, we could say it's non-contagious, but But when we talk about non-communicable, that means we can't spread them from person to person, okay? And by the way, when I'm talking about non-contagious, when I say they're not spread from human to human, they usually are spread through a vector. Okay, let me erase a little bit of this so I can make room for the rest of what I should have said about non-contagious disease. Non-contagious diseases don't spread from human to human, but they do spread to humans. They are communicable, but they are spread through what we call a vector. A vector is some kind of carrier that is non-human. Well, actually, a vector is simply a carrier. You can have human vectors, people that are carrying disease from person to person. But usually when we're talking about non-contagious, we're talking about spread through a vector that is not a person. For example, some diseases are carried by arthropods. So when we talk about an arthropod vector, we're talking about ticks. Arthropods are things that have external skeletons, like ticks and fleas and lice. They can carry diseases. Those are communicable diseases. They can be spread, but what happens is you have the disease and you're not going to spread it directly to another person, but if a mosquito starts to infect or bites you and then goes and bites another person, then, or a tick comes off of your body and then bites another person, it can spread that disease to someone else. So the fact that they're communicable means it can be spread to humans. If they're contagious, that means one person can spread it directly to another by breathing on them, droplets from coughing and sneezing. Non-contagious simply means that there's some other vector besides a person has to spread it. Arthropods can be vectors, and sometimes we can call certain animals. Like you can get rabies if a bat that has rabies bites a person or another animal infected with rabies, they can carry rabies to another human being. And whenever there's an animal vector, we call that a zoonosis. And you can think of zoos of animals. Zoonosis simply means it's an animal vector. If it's an arthropod vector, we're talking about ticks, fleas, lice, mosquitoes. And those can spread non-contagious diseases. Again, contagious means it's spread from human to human. Non-contagious means it's spread through some other vector that's not human. Arthropods, animals, and other things. And again, these are called communicable diseases because they can be spread to people. Non-communicable simply means that they're not going to spread to people. You can get them, but you would have to get them from something that's not taking it from person to person. Like if you eat contaminated food and you get food poisoning, that would be a non-communicable disease. It's not going to spread through the population. If you have food poisoning and you get bit by a mosquito and it bites your partner, they're not going to get food poisoning either. So that would be non-communicable. Now, some diseases can be described on how they impact a population or spread through a population. So here's some terminology that you should really be familiar with, okay? And this blue marker is messed up, I think. So we're going to talk about an outbreak. So when we talk about the outbreak of a disease, man, this marker's not working either. Let me get rid of that one and rewrite this. Oh, I'm glad, because it messes up my board. So when we use the term outbreak, what we're talking about is a sudden rise in the occurrence of disease or an illness in a population. And we could put above the endemic levels, which leads to another definition, okay? But when we have a sudden rise of an illness in a population, we call that an outbreak. Now, some diseases are endemic. When we talk about endemic, we talk about an illness that is common to an area or population. It has a regular occurrence. Okay? So, essentially, when we're talking about endemic, we're talking about this thing is always present in this area. For example, the common cold is endemic. It will stay in a population for long periods of time and occur very regularly. Or we could talk about malaria. Malaria is endemic to very tropical regions as it's spread by a plasmodium, by a particular organism. And so we can say that this is an endemic illness or an endemic disease, and that it's common to this area or population, and it's there regularly, okay? But if I have an outbreak, that means I either have a sudden spike of that, so I might say that, you know, a certain percentage of a population in an area is going to have malaria, and we're going to see a certain number of malarial cases in tropical regions. There might be, say, one person in every 1,000 might particularly have this disease normally. But all of a sudden I start seeing a bunch of cases pop up, and now for a period of time, it's not 1 in 1,000, it might be 1 in every 100 starts to develop the disease. We've had a 10-fold increase. Now we have an outbreak of that particular disease. Okay? And sometimes you can have outbreaks that are not of an endemic level. That's why I put this. The endemic level is the normal level that this disease or illness is within a particular population over a period of time. If you see spikes, that's a sudden rise in the occurrence of an illness. Now we have an outbreak. Okay? Now, you can have a disease that is not endemic to a population. Like, for example, meningitis is not endemic. It's not common in the United States. But at one time, we had a quick outbreak of meningitis cases, which is now why they force you to get, or highly encourage you, so that you can enroll in a college to get a meningitis vaccine, because it happened to have, there was an outbreak in a particular couple of dorms in the United States, and so now all college students are recommended to get the meningitis vaccine. Okay, now as we talk about endemic, another thing we can talk about is epidemic. Epi means uppermost or above. So when we're talking about an epidemic, this is an outbreak in a large population, or it's an infection or an illness that occurs and affects a large part of a population. So it affects a lot of people. A large number of people are infected or have had an outbreak. So now we say that this is an epidemic. And if we talk about the term pandemic, pan means worldwide. So a pandemic is an epidemic that has spread across the globe. For example, coronavirus and the COVID virus that we're talking about, COVID-19. There was an outbreak. It became an epidemic. It's infected large parts of the population, and now it's gone pandemic. It's spread throughout the world. And so those terms all mean something different, and you should know that. And by the way, some diseases can be described as being sporadic. This means they occur occasionally in a population. And usually when they're sporadic, they're very short-lived. A few people get infected and then it disappears. And then a few people get infected and then it disappears. So we call those sporadic infections. So an outbreak means a quick rise in the number of incidences of cases. Endemic means there's a certain number of cases that are natural to that population and are somewhat common to that population. Epidemic means that you have an outbreak that has spread to a large number in the population or a large part of the population, and pandemic means the epidemic is starting to spread worldwide. Sporadic are just diseases that occur occasionally. We see them pop up and then they disappear. Okay? Now, so that's one way that we can describe diseases, is, you know, their occurrence within a particular population. Another way to describe the diseases is how do they present themselves? When we talk about how a disease presents itself, one of the terms we talk about is acute. Acute means that there is a quick or rapid rise in symptoms. It is usually very short-lived. So if you have an acute infection, that means it popped up suddenly and it didn't last a long time. They develop very rapidly and are gone within a short period of time. Sort of like the flu. When you get the flu, bam, you get sick, it lasts a little while and it disappears. That would be an acute infection. A chronic infection means that it is slow developing, but can last a long period of time. So, when we're talking about chronic infections, one of the things that we can say is something like hepatitis B. Hepatitis B, when people get it, they don't get sick right away. It takes a long time for it to damage the liver and the signs and symptoms to appear from hepatitis, and you pretty much have it for a lifetime unless you get a specific type of treatment. Or another chronic infection would be HIV, human immunodeficiency virus. When people first contract it, they may not display the signs and symptoms of AIDS, but over time it will develop, slowly decrease your white blood cell count, and then once you have it, it's with you for a lifetime. There's no way of getting rid of it. So those would be chronic infections. Now, another term that we can use is subacute infections. And subacute infections are sort of somewhere between acute and chronic infections. The symptoms are less pronounced. The symptoms are not very pronounced. And they're usually not very long term. Okay? So, well, I shouldn't even say that. It's not that they're not long term. They are prolonged. Okay? I got that wrong. They're not very pronounced, but they are prolonged. For example, like what we call endocarditis. Endocarditis, the symptoms aren't really something that you notice really right away, or don't become a big problem, but it persists for a long time. Okay, we call those subacute infections. When we talk about a latent infection, in latent infections, it simply means that the symptoms don't appear right away. This means the infectious agent, sometimes my N's and my U's look the same. The infectious agent lies dormant for a period of time. So signs and symptoms only appear as the pathogen expresses itself. Okay? So a good example of this might be something like herpes, where herpes simplex 1, people get the infection, but it lies dormant in the body. And then something triggers it, and then the signs and symptoms can appear only when the pathogen is expressing itself. Herpes simplex 2 happens the same way, but only it expresses itself on the genitalia. So one is sort of the kissing disease, and the other one is a sexually transmitted disease. And so herpes simplex 1 is like cold sores. Herpes simplex 2 is genital sores. and those can be latent infections. They don't always express themselves immediately. If we talk about a non-fatal, fatal meaning death, fatal infections are infections that can lead to death. Non-fatal means they're not going to kill you. Okay, and you should know those two terms, non-fatal and fatal. I think everyone's familiar with those terms. So now, we can describe a disease based on how it affects a population. Is it endemic? Is it epidemic? Is it pandemic? We can describe how it presents itself. Is it acute? Is it chronic? Is it latent? Is it subacute? Is it fatal or is it non-fatal? We can also talk about infections based on the area of the body affected. For example, if I talk about a local infection is, what's the word I'm looking for? I'm not looking for contained. Local infections appear in a limited area of the body. Okay. For example, like the herpes virus, only herpes simplex 1 is expressed only in the oral cavity, and so it's a local infection. Appendicitis only occurs in the appendix. Pneumonia affects the lungs. STDs are unique to the reproductive tract. So those would be called local infections. Okay. If I talk about a systemic infection, this is an infection that can spread throughout the body or throughout body systems. So they don't just infect one tissue or organ, they can spread from organ to organ. And again, we mentioned this, I think, in a previous video. We've talked about sepsis. When your blood goes septic, that means some microorganism or toxin is in your blood and it's spreading all throughout the body. Those would be systemic infections, and they're not good. We talked about septicemia. That means you have sepsis in the blood. Bacteremia, which means you have bacteria in the blood. Viremia, you have viruses in the blood. Toxemia means you have toxins in the blood. And then we can talk about what's called a focal infection. A focal point is the area where the infection occurs. So the focal infection is the area infected by or affected by an infection. Okay, and sometimes this isn't used a whole lot. But when we talk about focal infections, we're talking about the specific area of the body that's affected by that infection. And we can say that this is a particular focus of the infection. For example, you might have a skin lesion, and that is where the infection is focused. Some focal infections can spread to become systemic. Like if you had a focal infection of Staph aureus in the skin, and you were trying to treat it, and then it gets into the blood, then you could say that that focal infection has now become a septic infection, or systemic infection, and result in sepsis. Okay. When we talk about what's called the pathogenicity, so I'm going to use two other terms, okay, and the virulence. So this is sort of, we're talking about here. The pathogenicity describes the likelihood an infection can spread. That's sort of the non-scientific way to think about pathogenicity. When I talk about pathogenicity, what I'm really talking about is like the capacity, the ability a pathogen will produce disease. What's the likelihood that you're going to get it? What's the likelihood that it can spread? How pathogenic is it? How many people is it going to infect? Or how easily is it going to spread? Something that's not very pathogenic is not really likely to spread a whole lot. Something that's highly pathogenic is something that's really going to spread. Like COVID-19 appears to be highly pathogenic. It's going to spread to a lot of people. So it has a high capacity or ability to produce disease and to spread. What's the likelihood that you're going to get it? Okay, the virulence of something, and this doesn't just refer to viruses, is the intensity of the illness. How sick will it make you? How sick will it make a person? How sick will it make one? So pathogenicity is what is the likelihood you could get it, and virulence is how ill will you get once you get it, okay? So when we talk about pathogenicity is how likely is it to spread or for you to pick it up. Virulence is referring to how sick will it make you once you have it, okay? Some of the factors that can affect a person's likelihood to pick up an illness. What's the likelihood that you can become susceptible to a particular pathogen? And this is based on a number of factors. One of them might be gender. Now I know we live in what's supposedly becoming a genderless society, and there are people that don't identify as one gender or the other, that are non-binary or transgender. But the fact remains that genetically, you're either male or female, unless you're a hermaphrodite that are born with some male and female genitalia. But even if you identify as the opposite sex, your physiology and your anatomy is either male or female. If you're female, you're much more likely to get a urinary tract infection than a male is. If you're infected by the human papillomavirus, well, males are not going to develop cervical cancer from it, but females could. So your gender can determine the likelihood that you can get an illness or express an illness and how bad that illness can get. Males can get the human papillomavirus and carry it. They can get genital warts from it, but they're not usually going to develop into a cancer. Where in females, those genital warts and the human papillomavirus has been shown to cause cervical cancer. Men don't have a cervix, males don't, they're not going to get cervical cancer. And the likelihood a male is going to get a UTI is much lower based on a number of factors. It turns out that meningitis is more likely to affect males than females. For some reason it spreads amongst males much more quickly. So there are some inherited traits that can determine whether you're likely to get an illness or not. One good example is with malaria. Some people have a trait, a gene, that causes their red blood cells to take on a unique morphology. As you guys know, our red blood cells tend to look like little discs. If I could slice it in half, they're called biconcave discs. But in some people, a few of the red blood cells will take on this shape, which is the shape of what we call a sickle, that long thing that the Grim Reaper carries. And so we call that sickle cell disease. And some people can carry the sickle cell trait, and they have the gene for it, but not necessarily develop sickle cell disease or sickle cell anemia. And if you have the sickle trait, you are actually not going to pick up malaria. You're much less likely to contract malaria. And that's why sickle cell trait is much more common in tropical regions of the world. So you inherit a genetic factor, a genetic trait, sickle cell trait, that decreases your likelihood to pick up malaria. Some things that can affect your likelihood to pick up an illness would be things like climate and weather. Certain disorders and diseases or illnesses are much more prevalent in the colder part of the year, like colds and flus, where in the warmer part of the year or warmer climates, we tend to pick up more of the tropical illnesses, things like malaria and things. So climate and weather can affect, and temperature can affect the occurrence or the likelihood of a disease to spread or be contracted. Sometimes it's age. One of the things that happens every year when the flu breaks out is people who are really young and don't have a very strong immune system, or people who are very old whose immune system is starting to wane with age, are recommended that they protect themselves and maybe not go out to a lot of public places or to large crowds. And so our age can affect the likelihood that we can pick up a disease or carry it. Lifestyle affects it. People who are IV drug users can spread a lot of diseases amongst each other. things like hepatitis, HIV, some other diseases. Well, and I'll stop with that. People who drink a lot, alcoholics, can damage their immune system and their liver and be more susceptible to hepatitis, particularly hepatitis B. Some things that can also affect your likelihood to pick up a disease is like fatigue, exhaustion. So when we go, our immune system, it requires a lot of energy for your immune system to fight off an illness. And so if you exhaust yourself, your immune system doesn't necessarily have the ability to fight off certain diseases and illnesses, and you're more likely to pick up or become susceptible to something. Nutrition affects it. If you have a proper and healthy diet, then vitamins and a proper diet can help your body, can help strengthen your immune system and your body's defenses. We can also look at things like whether you have a healthy lifestyle, whether you're someone who exercises and runs and works out, you're much less likely to develop disease or have a healthier immune system. So lifestyle, nutrition, age, weather and climate, temperatures can affect things, gender. Oh, and if you're immunocompromised, so people who have things like HIV or AIDS or have a compromised immune system due to chemotherapy or based on our age, then we can be much more susceptible to illnesses and diseases. Okay. Now, one last thing I want to talk about is, I think I'm going to stop there. This video has gotten long enough. I'm trying to keep them short. So, a couple of things that I want you to know are, you certainly need to know Koch's postulates. You need to know a couple of disadvantages to Koch's postulates, or the limitations of it. You need to know that you need to know the difference between symptoms of the disease and signs of a disease. You need to know what a syndrome is, and you need to know some ways that we can classify or describe diseases. We can do it on whether they're communicable or non-communicable. For communicable diseases, are they contagious or non-contagious? You need to know terms like outbreak, endemic, epidemic, pandemic, sporadic. You need to know if a disease is acute, if it's chronic, if it's latent. You need to know if diseases are local, if they're systemic, if they're focal. And you need to know the difference between pathogenicity and virulence. And then know some of the factors that can increase the likelihood that you might be able to pick up a disease or become ill. And because certain pathogens are opportunistic, they look for these situations, they look for it, but under these certain conditions, we are much more susceptible to them invading our body or causing illness and pathology and disease. Anyway, I'm going to stop there. There's one last thing we need to do. Or a couple of last things we need to do. We need to talk about the 7 challenges of how a disease or how a pathogen can cause disease and illness. We need to talk about the phases of a disease where you become ill, you express the illness and you feel the signs and symptoms, you start to heal and recover, and then we need to talk about endotoxins and exotoxins. But that's a whole nother hour, and I'm going to cut this one here and do a 3rd video. Listen, thanks for watching. I'm sorry for being a little bit sporadic, but this is done under some duress, so please forgive me. But I hope you learned something. I hope you had as much fun as I did, and I hope to see you in the next video. Okay? If you find this helpful, let me know, and hit the like button. Thanks for watching.